Hi, Christian. Thanks for joining us, Christian. Christian, is it right to say you're a researcher at the Max Planck in Tübingen?
I'm still a PhD student for
PhD student?
a very long time.
Yeah, that was what confused me. Okay, well, yeah, and Christian's been working on trying difficult things with pan-genome graphs for several years. In fact, I first met him when he came to a kind of workshop course hackathon that we held in Portugal on computational pan-genomics, and so he's been pursuing a lot of the same ideas for several years. So really looking forward to hear what you have to say today, Christian. So the floor is yours.
Thanks for the introduction, and of course, thanks for having me. As Eric already said, I basically dipped my toe in most of the things you can do in graph already. And there's one thing that we actually more or less finished by now. And by we, I mean Sebastian and me. Sebastian is also here today, and he converted the idea and the initial Python version of this tool into a Rust version that's actually performant and not painstakingly slow. So today I'm going to talk about what we call graphSV or pan-SV, and it's our idea of a reference-free variant caller on graphs. So just as a quick reminder, we all know that linear references are not ideal. And even when they started to sequence the first human reference genome 35 years ago, they were aware that this linear reference genome can't represent the population that it should represent. And now we are basically at a point where we can start producing those graphs, working with those graphs. And the group that I'm in mostly works in Arabidopsis thaliana and is involved in the Arabidopsis thaliana 1001 genomes project. And as part of this, we are building genome graphs for the species. And then we want to describe the population. And in order to do this, we want to look at the variation that is present in this population. And using the linear, the VCF approach is not ideal if you want to look at the full population. First of all, the second you align reads to the linear reference, or you produce a VCF file, you're losing the sequence context. You won't be able to tell what the read actually represents, what is the underlying feature from the locus this read originates from. It's very hard to detect nested variation, and it's challenging to detect larger variations that are larger than your length. So we started working with graph structures, and most of these things are actually present in the graph already. Variation is represented in the graph. If you build a graph from whole genome alignments, we are building our graphs using the PGBH pipeline or slight variations of it by aligning full reference genome assemblies against each other. And these graphs contain the variation as a feature of the graph. They explain nested variation, and you don't lose the sequence context. So if you annotate your newly assembled genome, you will be able to tell that a large variation contains transposable elements, genes, or whatever feature you are interested in. One thing that's important to note is when you build such graphs, it's the results that you get are very, very dependent on the graph construction method. If you enforce linearity, you won't be able to see repeats. If you don't enforce this linearity and compress the graph as much as you like, it's very hard to make actual sense from the graph because it's just a ball of nodes and edges. So you will need to find a balance of what you want to answer and the graph to build for it. So we built one of the graphs that we built as part of this project contains of 26 genomes. And when you first look at the statistics for this graph, you are able to see that roughly 65% of each of the genomes is actually shared sequence that's 100% identical for all of the genomes. Then you get the yellow part over here, that private sequence contained nodes that are not aligned to anything else. They might be alignable, but because we enforce a certain degree of linearity, they don't align based on the settings that we use. And then in red, you see part of the graph that is not aligned, not sequence that's shared by all the genomes, but also not private sequence. And when we just look at the graph itself, we see that 25% of the graph is shared between all. And then, of course, the private and what we call the soft core in yellow and in red is somehow inflated because it's not shared by all. Something that's pretty interesting every time we build a graph is basically looking at the saturation. And when we look at the core genome, we see that there's a certain saturation going on. There's not much more sequence being added to the core genome, while the pan-genome itself is more or less steadily increasing. And this more or less steady increase happens because the private variation keeps piling up, at least based on the settings that we are using for graph construction. And now we want to describe the variation that is present in such a genome graph. And we want to be able to describe this variation in a reference-free way, and in a way that's easy to access for non-bioinformaticians. So using file formats that most biologists know and can use. So what we do is we step away from reference and use the pan-genome itself as the reference basis. And we define bubbles in our graph based on sharedness in the graph, in the pan-genome, based on what we call the core level. That's something I'm going to explain in a second. The bubbles that we build are defined by shared anchor nodes. So they all the ways of getting through one of these bubbles have an identical left and right anchor node. And it's the same for all of them. And by doing this, we can then define a nested parent-child relationship between core or between higher level and lower level bubbles, and therefore represent nested variation in our data structure. So in order to explain what we are doing, I built a small tiny mock-up graph here, and we can already see that there's basically two haplotypes, the upper one and the lower one here, and both of them have some kind of variation. This whole graph has 5 paths, but these 5 paths originate from only 4 genomes. So we have genome 1, 2, with each one contact present, and then genome 3 is 2 different contexts. Those are the 2 blue paths. And then genome 4 with another. So once we start detecting bubbles using our algorithm in such a graph, we first do something that we call core level calculation. So for each node, we calculate how many genomes, not path, genomes, touch each of these nodes. And I indicated it here as numbers in the nodes. And based on this core level, we're going to define where bubbles start and bubbles end. And then when we start detecting those bubbles, we walk each path, starting at the leftmost node, going to the rightmost node. And each time we detect a change in core level. So from 4 to 3, in this time, we open up a bubble. If we go on, we can open up another one because it's from 3 to 2. But as we didn't close the first one, this node B here gets added to the nodes that is part of this first traversal. We can then, if we move on, close the first bubble because there's an increase in core level from 2 to 3, and then close the next one here at node E because it's 4 to 4. If we then traverse the next path, we can, because it uses the same nodes in the same order, we build the exactly same traversals for both of the variants that we've seen. If we go through the next path, the first path of genome 3, we can see that we basically open this large traversal from A to E as well, but we don't create a bubble here or a variant here because there's no change in core level. It's going from core level 3 to core level 3. So at the moment, we are blind to this presence-absence variation event. We go through the other 2 paths and detect the changes in core level there. And then the next thing is we merge traversals that are identical. So the green and the red path, they contain or traverse the same nodes in the same order, and we basically noted them twice. So we can then merge them into one traversal each, and then move on to construct the actual bubbles. And the bubbles are now constructed based on the start and the stop node. So all traversals that have the identical start and stop node are combined into one bubble. In this case, we get this large bubble from A to E, all path usage, and then we can note down what nodes are part of this bubble and what are the left and right. The same is true for this upper thing here. That's only used by green and red, and blue is still invisible for us. And this lower part down here from F to E as well. The next step is we now go back into the graph and detect those instances in the path where we have nodes that are anchors, left and right anchor of the bubble that are directly adjacent to each other in the specific path. So for example, this instance here where B and B, which are anchors of node 2 here, they are directly adjacent. This means there is a variation, but in this instance here, it was, in this case, probably deleted based on the frequency. And then the last step that we can do is we do a hierarchical clustering, and based on the nodes that are present in a larger bubble and in a smaller bubble, we can then tell if, in this case, this large bubble, bubble number 1, fully contains bubble number 2 and number 3, but they don't overlap, they are independent. So we get a bubble structure like this, which represents the nested variation in our line. We then output up to 4 files. The last one down here has to be requested using a parameter. The first 3 here are default files. And the very first one is a simple batch file. We have the normal batch structure, the sequence ID, start and stop of a variant, the bubble ID that this variant belongs to, and then the core level. So how many of the genomes or ecotypes in the graph traverse through this bubble. We then get another file where we pick up the node ID, the bubble IDs, and then we're able to tell how many parents this bubble has and children it has. So for example, for this bubble here, these are in the hierarchy higher bubbles, and those are nested sub-variation bubbles of this bubble. For each bubble, we then also introduce a statistic file that tells you how many traversals we have, how large the largest is, how small the smallest is, and some other basic statistics. And then something that can be requested and is, in my eyes, very interesting is an extension of the normal BED format, where in addition to the positions and the bubble ID and the core level, we also indicate the traversal ID. This means we are then able to tell for each variant which genomes that go through this bubble take the same route through this bubble. At the moment, well, not only at the moment, but it's something that will stay this way, there are certain limitations that you need to keep in mind if you want to use harness for GSV. We can only detect bubbles that are present in the graph. There's no realignment happening. It's a very simple bubble detection algorithm. And if your graph doesn't represent the bubbles that you would like to see, the variants that you would like to see, you won't be able to see it using this approach. Then complex variation with a lot of loops and paths traversing in and out can be, it will be detected, but it can be very challenging to interpret in the end or to know what's going on. And at least for us, it works best with the chromosome scale assembly. You need path in your graph. So an RGFA, for example, won't work. And then it's also best to have full chromosome scale assemblies. Small context might already be challenging. And this whole thing is still under development. We are still introducing new features. So one of the main things that we're currently working on is describing and categorizing the variants. So putting them into rough categories based on what has been used before. So categorizing things as SNPs, as presence-absence variation, maybe as double duplications based on statistics of this bubble. We are then interested in, and in the Python version it's already implemented, not in the Rust version yet, to add sibling information. So there are instances where there's no change in core level, but traversals from different unique independent bubbles use the same node space, the node subspace. And that's something that's biologically relevant, but it's hard to get this information from our output files. And this can't be represented in the strict parent-child model that we have. Therefore, we are introducing the sibling information where we are able to tell there is a subpart of this bubble that's also used in a certain other bubble. So the sequence is related, but not represented in a way that we would normally represent it. And then we also want to input or output repeats and inversions, but that's something that we haven't looked into yet, and that might actually be rather challenging. Now that you know how this whole thing works, let's see how it performs. So we built a total of 105 test graphs with 3 different linearity setting combinations using the PGBH pipeline and 6 graph styles. We always include the reference genome, in our case of Arabidopsis thaliana, and then different numbers of additional genomes. And the linearity is either a complex graph where we allow for a lot of repeats, so a lot of retraversals of a node by the same path with a lower sequence identity for the alignment and a low block length for the alignment, meaning we are able to compress more sequence in smaller nodes. We then build an intermediate graph and a fairly linear graph where we need a high identity for the block, a large block length, and a very low repeat count. You can already see if you look at this plot here that the node to edge ratio goes down in our graphs based on the complexity or the linearity that we enforce. On each of these graphs, we detect variance using graphSV, and then we compare it to 2 linear reference-based whole genome alignment structure variation detection tools. First of all, Siri, which uses Minimap2, and then based on this alignment, detects variation from this alignment, and Assemblytics, which is a pretty well-known tool. So the first thing to look at is the raw performance. So we see the runtime here, which is fairly linear. It's a graph with 1 genome plus the reference, 2 genomes plus the reference 4, 9, 14, and 19. And we see that even for a graph that contains 19 genomes, the runtime on one core is mostly under 15 seconds. And then the memory consumption is also pretty nice. It's not crazily inflated, although the Arabidopsis genome is pretty small. So 19 human genomes might already take up more space. And we compared our calls to the calls that the 2 linear reference tools produce. Oh, there's something wrong here on this slide, but you can still see the bar plot. And we see that, especially for Siri, the sensitivity, so how many of the features do we at least touch to more than 30%, is pretty high. And for Assemblytics is also pretty high. But if you look at the comparison of either Siri to Assemblytics or Assemblytics to Siri, they perform down here way, way worse than what we are able to achieve from the graph directly. And if we then look at the specificity, so not only do we touch this variant, but how much do we touch of it? We see that for Siri, we are in most cases pretty accurate. We almost touch 100%, cover 100% of the variant. For Assemblytics, it's slightly, or not slightly, but it's worse, but still it's manageable. And now coming to the end, I want to take you through of what we've doing so far with this tool. And in a small example, we are looking at a subset of 6 Arabidopsis genomes now and the reference genome. The second genome here is basically our reassembly of the genotype that has been used for the initial reference genome that is in use today. And then we have 5 additional ones. We produce genome annotations for them. And what we're seeing now here is the genome annotation. And in gray, we see genes that are assigned into ortho groups and don't change their position much. And in red and in blue, we see genes in author groups that are moving from chromosome to chromosome, that are inverted, and so on. Now we're looking to see and detect if we can find those genes and rediscover them as variants in our graph. And we have the small example here where we have one gene that is present in one copy in 3 other genomes and 3 copies in one of the genomes. If we look at how it's distributed, we can see that it's one copy, and here it's 3 copies. And if we look into an IGV view, we see the gene that is covered here in this instance by a structural variation that we detect using GSV, and it also covers a transposable element. And what we see now is this transposable element is jumping around with this gene in the genomes, and is present or represented as a bubble in the graph here. Next session, it's the very same. So we have our annotation. That's the ortholog of the gene that we are looking at. That's another gene that has been annotated, probably the transposable element gene. And once again, we see it's in the bubble with the very same feature here, that it's a repeated left and right anchor. That's probably a reminiscent of the insertion event of this gene that has been dragged along by the jumping tube problem. So that's basically it for a small tour of graphSV. A very big thank you to Sebastian, who recoded this whole thing in Rust and who have been working on with this tool. Big thanks to my boss and the rest of Weigel World, the Arabidopsis thaliana 1001 Genomes Consortium, and then what we call the happy graph people that are mostly here as well, that I had a lot of great discussions with. And if there are any questions, please go ahead and ask.
Thank you, Christian. Very interesting. Alternative approach to bubble detection. There are a few questions in the chat. The first one is Andrea asking, what is the computational complexity of the algorithm?
For now, it's linear. So as we traverse each path after each other, the more path you add, the longer the runtime gets. So it's scaling linear.
Okay, that makes sense. Fawaz asks why the algorithm is constructed this particular way. Instead of using known algorithms for bubble detection and super bubble detection, for example, Onodera et al. 2013, of course, that is an average worst case linear time and can be modified to return nestedness. There's also a tool called Bubblegun, which implements this, but it doesn't report the better SVs, but it does describe the hierarchy of bubbles. And so I guess the question in the abstract is, why did you choose to do a new approach when there are existing ones to detect bubbles? So
the Bubblegun approach is one that we've seen after we started this project. So the initial idea is more than a year old, the initial prototype. Then Bubblegun wasn't released. As for the other approach or approaches, to be honest, we started with it without actually knowing about them. And then we kept going and it's performing pretty well. So that's why we kept at it. Okay, that's very interesting. I wish I told you guys a bit more about this, but in any case, it's an interesting direction to do this. And so in particular, I was curious about the SV detection process for the orthogroup analysis. You said you used the method to find SVs. Would you describe that in a little more detail? Do you take the bed file that's being output? Is that the kind of input you need for that analysis? How does it work? So there are two inputs for this analysis. First of all, we annotate the genomes using different annotation tools, and then run orthofinder on those output GFFs to detect the orthogroups. And then we overlap each GFF file with our bed output file to see if we find genes that are located to at least 90% in one of our detected variants. And then we can check in the orthogroup or the orthogroup file if this orthogroup is something that we call a non-standard orthogroup. So an orthogroup where we have genes where we have different number of copies in each of the genomes, if we have copies that are located on different chromosomes. And that's how we basically overlap this. In the end, it's just a way to identify, at least for us, author groups and changes in author group or in gene number in author groups that are due to large structural variations instead of loss of a start codon or loss of whatever. Yeah.
I see. So I think maybe in response to Fawaz, that also indicates that you benefit a lot from having the bed type output against the paths of the pan-genome graph. So decompositions, bubble decompositions that could provide that for you would be equivalent, basically. They let you do the same analysis, right?
Yeah, yeah, for sure.
Yeah, and then I mentioned it, but then you said it, but it looks like the transposable element in this case had the target site duplication, which is a common phenomenon.
Yeah.
And then I'm curious if you were using the Rust handled graph library, if Sebastian's using that to implement the new version.
That's something you would need to ask Sebastian. Not 100%.
Okay.
But I guess he's in the chat. He should be able to answer.
That's fine. We'll ask offline.
Yeah, that would work.
Okay. I'm not seeing any other questions. We're about at time. So thank you, Christian. Really interesting. Don't quit hacking on pan-genomes. And yeah, thanks so much to you and all the other speakers for this really nice session.